Assalamu alaikum, dear students. My today's topic is photosynthesis. I hope you will enjoy the lecture, so stay tuned. Photosynthesis. This is a process by which the green plants manufacture their own organic food from simple inorganic substances like carbon dioxide and water in the presence of sunlight and chlorophyll. This is a very important process in the biosphere and it is an anabolic process. The generalized equation for photosynthesis has been written since earlier times, like 6 times CO2 plus 6 times H2O. This gives us C6H12O6 plus 6 times O2 in the presence of sunlight and chlorophyll. So this model of generalized equation of photosynthesis, this is the oldest model of the equation of photosynthesis. But this model is very much confusing and it has put confusions into the minds of plant physiologists. This is because when we take this model, here we write we have 6 times CO2 and 6 times H2. For manufacturing C6H12O6. You know that for manufacturing glucose, we require 12 atoms of hydrogen, which are very much present in water, 6 times H2. And also we require 6 times oxygen, 6 atoms of oxygen, they are also present in water. So what we require further in order to manufacture glucose molecule, that is this C6 we require. And this equation gives us a notion that the C6 comes from this carbon dioxide and it combines with water like this to make C6H12O6. The remaining 6 times O2, 6 times O2 comes out in the form of oxygen, that is 6 times O2. However, this is not a fact. The fact is that this oxygen which is evolved in the process of photosynthesis, it never comes from carbon dioxide, but it is always water that furnishes photosynthetic oxygen into the atmosphere. So this is wrong equation. That is why it has put plant physiologists into trouble for many years. It was C.B. von Neil, a bacteriologist, who observed that some photosynthetic bacteria manufacture their own organic food in the presence of light and in the presence of bacterial chlorophyll. And he was working with a photoautotrophic bacterium, namely Chromatium venosum. So we will take here C.B. von Neil. C.B. von Neil. He in 1931 performed an experiment with a phototrophic bacterium that you call as Chromatium venosum. Chromatium venosum. Photoautotrophic bacterium. He observed that this photosynthetic bacterium manufactures glucose and utilizes carbon dioxide and a hydrogen donor, that is hydrogen sulfide, not water. So in the process of its photosynthesis, it does not evolve oxygen, rather it evolves sulfur. So he wrote the equation for the bacterial photosynthesis as 6 times CO2 plus 12 times H2S in presence of sunlight and a pigment called bacteriochlorophyll. Bacteria have their own type of chlorophyll molecule, that is bacteriochlorophyll. And he observed that it manufactures C6H12O6 plus 6 times H2O plus 12 times S. So you see that in this bacterial photosynthesis, as per this equation written by von Neil, no oxygen is evolved, rather sulfur is here released. And he called this as an oxygenic type of photosynthesis. This is said to be an oxygenic photosynthesis. Why an oxygenic? This is because in this photosynthesis no oxygen is evolved into the atmosphere. Okay, so 6 times CO2 plus 12 times H2S in the presence of sunlight and bacteriochlorophyll gives us glucose, water and 12 times S. It is the equation of bacterial photosynthesis, and von Neil tried to write the photosynthetic equation on this model. He wrote the equation of photosynthesis of green plants based upon this model, that is he took 6 times CO2 plus 12 times H2O, sunlight and chlorophyll, plant chlorophyll. This gives C6H12O6 plus 6 times H2O plus 6 times O2. So this became the modern model of equation of photosynthesis. And in this model, you see here we have 12 times water rather than 6 times water. And basically this carbon dioxide is to be reduced so that it can manufacture C6H12O6, but for that purpose it needs to be reduced by water. Water provides the necessary protons and electrons for reducing carbon dioxide so that sugar will be manufactured. And from this water, oxygen is liberated out as molecular oxygen. So this is the oxygenic type of photosynthesis. This is oxygenic type of photosynthesis which is performed by all the green plants, and this is also performed by cyanobacteria, blue-green algae. Okay, so this is the generalized equation for photosynthesis as per von Neil. Now, how can we be sure about the fact that water is the source of oxygen in the process of photosynthesis? For that purpose, many scientists came from time to time and they gave their models, and experiments were carried out also in order to prove that oxygen evolved in the process of photosynthesis comes definitely from water and not from carbon dioxide. Among those experiments, we have an important experiment that was performed by Robert Hill. Robert Hill in 1937 and also later on in 1939. This experiment was conducted in pots, and it was Robert Hill who performed this experiment to prove that water acts as a source of oxygen in the process of photosynthesis. He took isolated chloroplasts of a plant which is called as Chlorella media. Very small plant, Chlorella media. This is a herbaceous plant, very small and very common in our orchards, in the lawns and along the roadsides. It has a small white flower, Chlorella media. It is isolated chloroplasts were taken. Suppose I will draw here a beaker. This is the beaker. And in this beaker he kept water. Water को ले लिया इसने. And he put isolated chloroplasts of Chlorella media. This. The chloroplasts were isolated carefully and put in the water. So he made a suspension of the chloroplasts in water. And he also illuminated this. So light was made available. Light was made available. And also he put an oxidant, oxidizing substance. That is a substance which can receive the electrons, which can accept the electrons. And he took many suitable electron acceptors which are said to be Hill oxidants. The Hill oxidants are many that were taken by Robert Hill. Let me tell you about those Hill oxidants. So I am writing here Hill oxidants. Number 1, ferricyanide. Ferricyanide is an important Hill oxidant. Number 2 is ferrioxalate. Ferrioxalate. Number 3 is chromate. Number 4 is benzoquinone. Benzoquinone. And also there is one more, that is DCP-IP. DCP-IP stands for dichlorophenol indophenol. Dichlorophenol indophenol. Basically this DCP-IP is a blue dye. Blue dye, ये एक रंग है, dye है. And it is blue in oxidized state. ये साथ ये आयसी oxidized. जब ये oxidized होगा, and in that oxidized state it is bluish in color. So oxidized state में होता है ये क्या? Blue. This is a list of the Hill oxidants. And now let us try ferricyanide. Ferricyanide was taken by Robert Hill and it was put in this suspension of chloroplasts that were suspended in water in the presence of light. And he carried out a reaction that you call as Hill's reaction. उसी को हम photosynthesis में क्या बोलते हैं? It is called Hill's reaction. Hill's reaction. इसको आप समझिए please. The Hill's reaction has proved this, that water can act as a source of oxygen in the process of photosynthesis. So, in order to write the Hill's reaction here, the equation for that Hill's reaction, I will write here 4 times FeCN, ferricyanide, whole 6, 3 negative. That is, the oxidant is in an oxidized state. This is ferricyanide, and we will take twice H2O. This reaction, you know, it is occurring inside beaker and immediately inside isolated chloroplasts in the presence of sunlight and isolated chloroplasts. Isolated chloroplasts suspended in water. And after some time, when sunlight performed its role, chloroplasts also performed its role. These two reactants, that is the Hill oxidant and water, they performed their own roles and carried out this reaction. After some time, what was produced? Hill observed that there is production of a reduced substance, that is FeCN6, whole raised to power 4 negative, plus twice O2. Sorry, not twice O2, it is O2. I'm sorry. Plus O2, that is oxygen, plus 4 protons. 4 protons. This water was split into 3 substances or products. One is the oxygen, another product is the protons. 4 protons were liberated out, and 4 electrons. The 4 electrons were received by this substance, that is ferricyanide, because it is indicated here. Here you had how many free electrons? 4 3s are 12 electrons. And here you see we have 4 into 4, that is 16 electrons. So 4 electrons are more in this substance than this. And these 4 electrons, which are extra, they came from water, 2 molecules of water. And they were accepted by this ferricyanide, and ferricyanide got reduced. And this is the Hill's reaction. This is said to be Hill's reaction. So Hill's reaction demonstrates that water can act as a source of oxygen in the process of photosynthesis. Because in this Hill's reaction, there is no carbon dioxide. When no carbon dioxide is present, this means that only water is present and water gave out oxygen. So this indicates that water can act as a source of oxygen in the process of photosynthesis. Okay. Then another experiment was carried out by some workers, namely Reuben, Hossid, and Kamen, in 1941. These 3 workers took a green alga belonging to class Chlorophyceae. The alga is Chlorella. You may be knowing Chlorella, a unicellular green alga, spherical in shape, non-motile. This is covered by means of cell wall. Internally, it has a living protoplast and it contains a cup-shaped chloroplast. So this is the structure of Chlorella that was taken by these 3 workers. So Chlorella was made to perform photosynthesis in the presence of carbon dioxide and water. But the water that was taken, it had a labeled oxygen isotope, that is O18, heavy isotope of oxygen. You know that normal isotope of oxygen is O16, but they took water which contained oxygen with O18 isotope. So they wrote the equation 6 times CO2 plus 12 times H2O18. So oxygen is labeled radioactive isotope of oxygen that is associated with water. This water was employed to carry out the process of photosynthesis in chlorella. So here we will write sunlight and chlorophyll. What they observed? They observed that there is production of 6C6H12O6 plus 6 times H2O plus 6 times O218. Radioactivity was checked in the products. Radioactivity was checked whether it is present in sugar, it is present in water, or it is present in the molecular oxygen that is evolved. And it came to be associated with that oxygen which is evolved, that is this. It was found to be radioactive. So, had oxygen been liberated by carbon dioxide in this process, then this oxygen would have been found in what? It would have been found here. Had carbon dioxide been able to produce what? Free oxygen in the process of photosynthesis, then the liberated oxygen wouldn't have been radioactive isotope. So this clearly indicated beyond any doubt that the source of oxygen in the process of photosynthesis is water and not carbon dioxide. Carbon dioxide नहीं होता है source oxygen का, rather it is water. So these were the various experiments that led to the discovery that during photosynthesis oxygen which is liberated in the green plants that comes from water and not from carbon dioxide. Okay. Now you know that for carrying out the process of photosynthesis there are supercialized cells in the plant. And these supercialized cells in the plant are said to be chlorenchyma cells. Chlorenchyma. Or we can say chlorenchymatous cells. Chlorenchymatous cell is a type of parenchymatous cell, living cell. And these chlorenchymatous cells in the leaves are said to be mesophyll cells. They are also called as mesophyll cells. These are restricted to green regions of the plant body. That is in the leaves, in other green plants of the shoot system, and they are never found in the roots. However, there may be some exceptions where the roots will be exposed and sometimes they develop the chloroplasts. But I tell you, chlorenchymatous tissue or chlorenchymatous cells, they are restricted to the green regions, especially restricted to what? Leaves of the plants. And their job is to carry out the process of photosynthesis. Inside the cytoplasm of a chlorenchymatous cell or mesophyll cell, there are supercialized plastids. And those supercialized plastids you call as chloroplasts. A chloroplast is bound by means of two membranes, the outer membrane and the inner membrane. So this is the chloroplast. And this chloroplast acts as the site for photosynthesis. It will carry out photosynthetic process. Photosynthesis. So where does the chloroplast occur? It occurs inside the cytoplasm of chlorenchymatous cell or mesophyll cell. Chloroplast is bound by two membranes, outer membrane and inner membrane. And there are two enclosed spaces, the outer space called as periplastidial space And the inner space, this is the inner space, and this is filled by means of a ground substance or matrix or stroma. Stroma, it is the ground substance that is contained within the inner space of the chloroplast. Stroma contains water, it contains inorganic solutes, organic substances, proteins, and enzymes. Almost a dozen enzymes are present in it. There are coenzymes like NADP, or there are coenzymes like FMN or FAD. These are various types of coenzymes which are present in the stroma. And there are also the rings of DNA, that is like plasmids, circular DNA rings, and they contain the genes. You know that chloroplast is a semi-autonomous body. That is why the plasmids are there, or circular DNA molecules may also be found. The ribosomes that occur in the chloroplast stroma are 70S type of ribosomes. And there are some RNA molecules also. Okay, this is the stroma. Now, in this stroma, there are stacks of thylakoids. And those stacks of thylakoids you can call as grana. A stack of thylakoids is called granum, and grana is the plural term for granum. So I have drawn here these stacks called grana. As so many grana are there. So actually, what is a granum? Granum is a stack of thylakoids. This is one thylakoid. It is upon another thylakoid, and so on and so forth. So when thylakoids get stacked together, they form a granum. Now the question is, what is thylakoid? What is meant by thylakoid? Thylakoid is actually a membrane. It is a membrane which is said to be a living membrane, made up of phospholipids and proteins. As usual, जैसे होते हैं सेल मेम्ब्रेन्स या एंडो मेम्ब्रेन्स, उसी तरह के होते हैं membranes of thylakoids. So a thylakoid, I am drawing here a thylakoid. This is not chloroplast. This structure that I am drawing here, it is actually a membrane. यहाँ से लेकर के यहाँ तक ये जो है membrane है, ये thylakoid membrane. This is the inner edge of this thylakoid membrane, and it encloses a small space which is called as loculus, or you can also call it as thylakoid channel. Okay, the inner edge lies towards loculus. Then this is the breadth of this thylakoid membrane, and this is its outer edge. Outer edge. It is bathed externally by stroma. यहाँ पे क्या है? Stroma है. So this is outer edge of the thylakoid membrane. This is inner edge of the thylakoid membrane, and this is the width or thickness of the thylakoid membrane. This width of the thylakoid membrane contains the phospholipid bilayer and the proteins as a constituent of this membrane. All the photosynthetic pigments, whether chlorophylls or carotenoids, they are present in this membrane. The pigments are present in it. So if you are asked where does the chloroplast contain or hold the photosynthetic pigments, your answer must be that the pigments are held inside the thylakoid membranes. They are not present in the stroma. The pigments are never present in the stroma. They are rather present in the thylakoid membranes. And these thylakoids become stacked to form the grana. The neighboring grana are interconnected by means of stroma lamellae or frettes. Suppose here extensions are present. These extensions are said to be the fratus or stroma lamellae. Fratus or stroma lamellae, or you can call them also as long thylakoids, or you can call them as agronal thylakoids because they are not present inside the grana. So this is the structure of the chloroplast. Now, if you treat the chloroplasts with a mild detergent, 4 types of supramolecular complexes can come out. They can be released. When you treat the chloroplasts with a mild detergent, the grana are disrupted by this activity, and what happens, the 4 supramolecular complexes that are contained within the thylakoid membranes, they come out. Now let us see what are those 4 supramolecular complexes. Four supramolecular complexes that are present within the thylakoid membranes of the chloroplast. Number 1 is the pigment system or photosystem first. Pigment system, or it is also called as photosystem first. Number 2 is the pigment system or photosystem second. Number 3 is cytochrome B to F complex. Number 4 is F0 to F1 complex. So these are the 4 supramolecular complexes that come out when we treat the grana or thylakoids with a mild detergent. In addition to these, there are several other agents present inside the thylakoid membranes, and important among them are quinones. For example, we have an important quinone, plastoquinone, which is a mobile electron carrier. It moves inside the thylakoid membranes. And how does it move? It moves from outer edge of thylakoid membrane to the inner edge. If you see here, this is the thylakoid membrane. And I told you earlier that this space and